Modification of Mendelian ratios, Part 2. Okay, now let's take a look at X-linked recessive traits. So the sex-specific pattern that you see with the inheritance of X and Y chromosomes, right? The X being given from the father to daughter and the Y being given from father to son. What that does is that helps us, well, it produces a distinct pattern of inheritance for X-linked recessive traits. So just like we saw with X-linked dominant traits, all affected daughters, no affected sons, we saw that. Same thing goes since we're dealing with a sex chromosome. There is still another distinct pattern of inheritance. Now, it won't be all affected daughters, no affected sons, but there will be a specific type of pattern of inheritance. So let me just show you what that is. So let's start with hemizygous males. So first off, they're hemizygous because they only have one X chromosome. So they're never going to be homozygous. They're never going to be heterozygous. They are always going to be hemizygous because they have one X chromosome. So hemizygous males that have a particular X-linked recessive allele are going to have an affected phenotype. And then only homozygous females are going to have that same phenotype because it is X-linked recessive. So you have to have two recessive copies. You can't have one of those copies of that, one allele of that gene on the X chromosome being the dominant allele, because then that would mask the recessive allele. In order to get the recessive phenotype, you have to be homozygous if you're female, if you're XX female. And so those are who are going to be affected. And so you can see the next bullet point, phenotypic expression is much more common in males than females. So that's the one that is highlighted. If you're making flashcards, like I said before, this would be the one where you put a little star by it because you know that it's going to be sex related, not autosomal, because you're going to count up males and females and you're going to see it skewed towards one sex. In the case of X-linked dominant, it's skewed towards females, where you see more females affected than males. In the case of X-linked recessive, it's going to be skewed towards the males. So like that's what this is. Phenotypic expression is much more common in males than females. And the affected males receive the mutant allele from their mothers. And then they transmit it to all their daughters. So they're never going to get the affected allele from their father because he's what gave them, the affected male, he's what gave them the Y chromosome. So they have to get it from their, affected males have to get it from their mother. And so that tells you something. If you have an affected male, that means that his mother is at least a carrier. If she has the phenotype, then she's obviously homozygous for it, and so definitely easy to spot. But if she is not showing that phenotype, the affected phenotype, well, we know she's at least a carrier. And so affected males transmit, or sorry, they receive it from their mother, and they transmit it to all of their daughters. So they never transmit it to the sons. Same thing. They transmit that one X chromosome that they have with the X-linked recessive allele to all of their daughters, 100% of their daughters, and 0% of their sons. Because if everything is happening according to plan, then the sons receive the Y chromosome, the daughters receive the X chromosome. And so, therefore, add this up, daughters of affected males are usually heterozygous. So they very well could end up being homozygous recessive, but that requires the mother to also be a carrier. The one thing we know is that at the very least, daughters of affected males are going to be at least heterozygous. And if they're heterozygous, well, then they're unaffected because it's an X-linked recessive trait. But then if you look at their sons, so next generation, their sons have a 50% chance of being affected. If you have an unaffected heterozygous carrier female, her sons have a 50% chance of being affected because she's a carrier and one of her X chromosomes is going to make it into her son, and therefore there's a 50-50 shot that he's going to get the one with the X-linked recessive allele and therefore show the phenotype, or the one that's not. If she's carrying a wild type allele on the other X chromosome, perhaps he gets that one. So here is a massive pedigree. There are some busy people on this. And so, again, it's not perfect, right? We don't have people crossed out to indicate that they're deceased. But we have some very busy people that had 1, 2, 3, 4, 5, 6. Wow. I don't even want to think of that, but it's 8, right? So 1, 2, 3, 4, 5, 6, 7, 8. So 8 children, 2, 4, 6, 8, 8 children. Wow. And then we have something very interesting. So, let's see. So here we have twins, right? And then where was it? There was somebody. Oh, right. I was going to say that's interesting is that he, by chance, had twins with her, and then a son, and then later had twins again with her, right, with her. And then also, nope, that's it. So, but anyway, I'm getting, into the gossip of this, so apologize. So if you just look at this pedigree, there's some easy things to look at. It makes it very easy. Don't get intimidated by the large size of the pedigree. Just look how simple it is, right? So, do you see something? Do you see somebody darkened in every generation? No, right? So therefore, we are already done with the whole dominant recessive thing. It's going to be recessive. Now there are some, if you do the questions in the back of the book, you will run into some exceptions, right, where they start throwing things that I'll mention to you in a few slides, things called penetrance and expressivity at you. But I wouldn't do that on an exam question. So basically, if you're looking at it and you see it skipping a generation, it's recessive. And it doesn't just skip one whole generation. It can, for example, here, so we skipped a generation to get to here, and then we skip another generation to get to here or to here. So we're skipping generations, right? And so if you're skipping generations, if that's possible, then you're dealing with a recessive trait. Then you want to know, is it autosomal or sex-linked? And so what you would do is you would count up the number of males and females. And so let me count up the number of females, 0. That was easy. And then we can count up the males, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. It doesn't really matter because it's heavily skewed. It's 100% skewed towards males. So that tells us that it must be somehow sex-linked. So now we are basically saying, okay, well, it's either going to be the only sex-linked options we have now are X-linked recessive, because there's no such thing as Y-linked recessive, and there's no such thing as mitochondrial recessive. So basically, it's X-linked recessive. That kind of tells you just by process of elimination. But of course, you also want to look for your bullet points, right? And so phenotypic expression is much more common in males than females. Check. Affected males receive the mutant allele from their mother and transmit it to all daughters, never to sons. Well, we don't have any daughters being, transmitted, but we do see that, okay, so he absolutely passed it on to his daughter and his daughter and his daughter. And so you can see that she's a carrier because he's not affected. And so these guys only got their X chromosome definitely from their mom. And so she's a heterozygous carrier. And then you have a 50-50 shot, and he got the mutant allele. And then next pregnancy, he got the mutant allele. So, it's a 50-50 shot each time. And then we could see that she is a carrier. And so she gives, okay, so 50-50 shot, right? So didn't get the mutant allele, got the got the dominant X chromosome, the one with the allele on it that was dominant, so the X chromosome containing the wild type allele. He also got the wild type allele from the X chromosome, and then he got the other X chromosome from mom, the mutant X chromosome that contained the X-linked recessive trait. So you can kind of you can see what's in there. And so now we can be pretty confident that this is an X-linked recessive trait. So let me give you an example of an X-linked recessive trait. So color blindness, that's why the slide went to grayscale. So color blindness is an X-linked recessive trait. It's much more dramatic in class. And so red-green color blindness affects 8% of males in the U.S., which is a large number of people. That's a large number of males. That's why you'll notice, since you can see the difference here. So you know how I use color to indicate, to emphasize certain terms and whatnot. Well, what you may not have noticed is, if you're just focusing on the color, is that I also do a shadowing effect. It's just one of the text effects you could use. So I do color and then I also shadow. And so that way, if you were to be colorblind, you could still see the difference. You could see right now. You can see the difference between this one seems to be highlighted, whereas with the shadow, whereas this one is not. And so that's how those terms are both bolded, shadowed, and colored, how they stand out regardless of whether you can see the color or not, because of course I'm going to have quite a few students that are colorblind. Now, when I say colorblind, remember I mean red-green colorblindness. Blue colorblindness is autosomal, that's something else. Red-green colorblindness is what we're talking about, and that is an X-linked recessive trait. So here it is. Red-green colorblindness is inherited as an X-linked recessive trait. As I just said a moment ago, blue colorblindness is an autosomal dominant trait. So that's a separate thing. That's completely different. All right, so if you want to, take a quick little test to see whether you have any level of red-green color blindness, for those of you that can see color, you can see, oh well, at least red and green. You can see the fall foliage changing colors here in this image, and then this is what it would look like. So you certainly can make out the yellow because that's not an issue, but the red versus green just appears pretty much the same to an individual with red-green color blindness. And of course, there's all sorts of tests you could take online where you could look at images, and hopefully if you look at this image, you could see if you are red-green colorblind, then you wouldn't be able to see certain insets, whereas if you are not red-green colorblindness or suffer from red-green colorblindness, then hopefully you can see, well, I usually ask my students what they see, and I give them a little time to answer the question. And so we get some people that say that they could see a 29 in here. And then my question to you is, what else can you see? Do you see anything? Right? Do you see that there are some I'll give you a hint. There are some very slight differences in the shades of green, right? So this would be like, I don't know, what do you want to call that? A drab olive, whereas this is going to be, oh, let's go with seafoam, right? I don't know. But the point is, it's got more of a sort of a turquoise. It's not quite turquoise. Anyway, I'm losing it. Do you see what else is in there? So some people at this point in the lecture usually say, well, I see like a maybe kind of like a 71 in there or something like that. And then I just say that I'm messing with you. There's nothing else in there. And that's true. It's just 29. The extra colors are for you to know whether, the degree of red-green color blindness, like perhaps if you can't see the 29, can you still make out some of these other things in there, like these other different greens? But it's 29. Those are there to kind of throw you off from the 29. So I tend to, unfortunately we don't have that dynamic over a recorded lecture, but I definitely tend to mess with my students because, it's amusing for me at least. Okay, so the genes responsible for the three forms of color blindness, as I said, blue is going to be autosomal. That's on a different chromosome, but the gene responsible for the production of the cone cells that detect wavelengths of light in the, so the electromagnetic spectrum of visible light that is red and that is green, those are going to be in retinal cone cells. Sorry, I cut myself off there with the slide transition. What I was saying is that those are going to be encoded by the X-linked recessive allele. So color blindness, red-green color blindness, is an X-linked recessive trait. And so you can see that kind of here. Here we have normal color vision female, so she's homozygous dominant. Here we have a colorblind male, right? So there you can see the color blindness. And so then in the next generation, what we can see is that we are going to, if they have a daughter, then she is going to be a normal color vision female, but she's a carrier. If they have a son, then he's free and clear, right? Because he's going to get either this one or this one. He's not going to get that. And so it will end with him. And so basically it's not going to be passed on any further in his family lineage, the color blindness that his father has. So if there are any males in our class whose father is colorblind, you probably aren't going to have red-green color blindness, and now you know why. It's because you got your, there's a little pun there. So you had the Y chromosome from your father, as opposed to an X chromosome containing a color blindness X-linked recessive allele from your mother. Now, if your father is colorblind and then you, as a male, are also colorblind, well, that's just, it turns out that means that your mother is a carrier. So here's another scenario, right? So colorblind female, normal color vision male. So now because of the male's normal X chromosome, the daughters are going to be normal color vision carriers. But then if you have a colorblind mom, a red-green colorblind mom, then her sons are definitely going to be colorblind, right? They're either going to get this one or this one, doesn't matter. They're both she's homozygous recessive, so they're going to get one of those. So, and there are other things that are X-linked recessive. I'm just using color blindness because it's, people have heard of it. But for example, white eye color in Drosophila, so in fruit flies, is another example of an X-linked recessive trait. And so, like if you buy bananas and leave them on the counter for a little while during the summer, and then you get you get some fruit flies. So, look closely at them before you kill them. And I shouldn't say that. It's funny because I was having a conversation with my student on how careful I am with the different organisms in a laboratory. I've worked actually in a Drosophila laboratory before, and how careful we were to try and treat them with respect and dignity. And these are fruit flies, so some of you might be laughing at that, but you don't want to cause any undue stress on any living organism that you're working with in a laboratory. And so we would do everything we could to basically, like if we wanted to take a close look at one, we would basically kind of anesthetize them, just kind of knock them out, and put them under a microscope, look, just like kind of move them around and stuff. Nothing dangerous, nothing major, nothing harmful. And then once they start to come back, you basically put them back in the jar so that they can go back to flying around. And so we were very, very careful with them, and we took so much caution. And then at home, on the other hand, I'm basically like a mass murderer of fruit flies because I hate them. I guess that's a little strong. I shouldn't say I hate them, even though I hate them. And so, I basically have, scientifically found a way to eradicate them from my kitchen. And so you can use apple cider vinegar and oil, or soap. Actually, you can use soap as well. And you can mix the two and put them in a jar, put a little saran wrap on there and poke a couple holes, and they'll be attracted to the inside of the jar for the apple cider vinegar. But then once they get in there, they can't get out. And so it's actually really useful to capturing them. Because, of course, my poor little children, once we had our little fly soup and all the little dead flies, we did have a couple in there that had white eyes, and so I started talking to them about the white eyes. And they were about, I think, 4 at the time. And so we didn't get past the ew, like they were just screaming at the little dead bugs. So I didn't really get a whole lot of genetics across to them at that point. But, I tried. All right, so now let's talk about if you do have 2 X chromosomes, like a female, XX female, and a male only has 1 X chromosome. The question is, do you use the same amount of gene product off of 2 X chromosomes in a female as 1 in a male, or does the female use twice as much X-linked gene product than the male because she has 2 X chromosomes and he only has 1? What's going on there? And so it turns out, so here, let's start with the beginning. So females have multiple X chromosomes, males only have 1. And let's see, females do not produce twice the amount of X-linked gene product as males. So I'm giving you that, right? So I'm answering that question. So females do not produce twice the amount of gene dosage, right? So X-linked gene dosage. So what they do is they produce the same amount of X-linked gene dosage, the amount of product, the amount of usage of their genes on the X chromosome is the same as a male with only 1 X chromosome. So we can envision a couple scenarios here, like how do they do that is the next question I'm about to ask. And so let me ask that question. How do they do that? Do they use both X chromosomes, but each one only about 50%? Whereas a male just cranks it up and uses his 1 X chromosome 100%, so that adds up to the same amount of gene dosage. Do they, use 1 X chromosome and then sometimes use the other X chromosome and then just alternate back and forth? Do they use 1 X chromosome, shut down, and get rid of the other X chromosome? There are some different scenarios that are possible here, and so that's the question. How is it that an XX female has the same amount of X-linked gene dosage as a XY male? And the answer is the title of the slide. It's called dosage compensation. The XX female has to compensate for the lack of gene dosage in males if they are going to both have the same amount of gene dosage. And so the way in which we learned about dosage compensation is quite interesting. So it turns out men and women produce the same amount of X-linked gene products. And so dosage compensation is the mechanism that I'll tell you about that regulates the expression of sex-linked gene products such that men and women produce the same amount of X-linked gene product. There we go. And so before dosage compensation was elucidated, there was also, there was an observation made. And so that observation was bar bodies. And it wasn't known at first what bar bodies were. But then later it became known that inactivated X chromosomes appear as bar bodies in cells. So if you're looking at these nuclei of cells that have all been stained, what you could see is that, okay, so here we have a 46 chromosome containing XY male. And to the trained eye, I don't expect you to be able to pick these out, but to the trained eye, you would see that there are no bar bodies in the XY male's nuclei in any of his cells. A 46, which is the normal chromosome number, right? 23 pairs of chromosomes for humans. So a 46 chromosome containing XX female has one bar body, indicated by the little white arrow. So there's a bar body, there's a bar body. The bar bodies are tightly coiled up X chromosomes that are so tightly wound up. They're basically like, imagine a piece of paper with all sorts of writing on it that you could read is a chromosome, and it has all that information on it that you could read. Well, if you crumple that up into a teeny tiny little ball, you can't get at any of that information. Think of that as a bar body. It's the X chromosome that's been tightly wound up, coiled up to the point where there's no access to any of the genes on that X chromosome. So much so that the dye that's used can't even really get in there. And so it creates this little light spot. So those are your, so there's a bar body. Here's a different nucleus from the same, 46 chromosome XX female, one bar body. Then if you're thinking, I wonder if these X chromosomes are what make up the bar bodies, well, then you could take a normal healthy cell and you could cram a couple extra X chromosomes in there. So this is done in vitro, but you have now a 49 chromosome containing cell. And you can see that its sex chromosome complement is 1, 2, which would be normal, 3, 4, 5. So there's the extra 3 chromosomes that got us up to 49. So there are 3 extra X chromosomes. Then if you look at it under the microscope, you can see 1 bar body, 2 bar body, 3 bar, 4 bar bodies. So that would be 1, 2, 3, 4, leaving you with 1 functional X chromosome, just like an XY male cells. Nucleus has. And so same thing here, 1, 2, 3, 4 bar bodies. So if you look at lots and lots of other micrographs, you'll see the same thing. So it turns out that these inactivated X chromosomes are these bar bodies. At least that's what the thought was. But nobody really had a good explanation as to why that is. Why would there be bar bodies in these cells? Then we get to Marie Leon's work. And so she's the one who made a link between bar bodies and dosage compensation. And so this actually goes back to our very first slide of this lecture. If you look at the title slide, you'll see some mice of varying coat colors in a litter. And so they're all litter mates. And so what she was doing was she was working on the inheritance of coat color in cats and mice and other organisms, but basically she started with mice and then worked her way up to cats. And so we're about to talk about why calico cats are generally female, pretty much always female, but basically mice and cats. And so her observations led her to provide a hypothesis for what is now known as dosage compensation. So I'm cutting to the chase there by saying she got it right, but she was trying to figure it out at the time. So here's what she saw. She saw that female mice heterozygous for an X-linked coat color. So this is dorsal coat color, not ventral, right? So this is basically along the back of the animal. If you were to breed certain mice of certain colors together, if you had heterozygous males or heterozygous females, you'd get different outcomes. So the female mice that are heterozygous for an X-linked coat color gene had unique coat colors. They had patterns. They had very unique coat colors, whereas the males were a solid coat color. And so the other thing is they would not have the same pattern or coat color as either parent. It wouldn't be a blend of parental coat colors either. It would be a completely unique pattern every single time. So you could apply this to mice. You could apply this to calico cats. Same thing, exact same thing that's happening in those organisms. And then we'll actually talk about some examples of what's happening in human females as well along these lines. So what she saw was that rather than like a blend of the coat colors, the fur had random patches of the 2 parental colors. So here you can see that this was a cross between black and orange, and you have, areas of black, areas of orange, areas of black, areas of orange, all over the place. And so usually every quarter I have my students name the cat, and I throw out there that patches is not an acceptable name. That's just not creative enough. And so my favorite quarter was, just this voice came from the back of the room and said, Steve. And so I was like, you do know that it's a female cat, right? We're talking about female cats. He's like, yep. And so I'm like, okay, Steve it is. We named it Steve. And I said, I have to ask, why Steve? And he's like, because I'm Steve. And so we get all sorts of names. We've had Bubbles. We've had Mittens, even though that one, I don't see that. But anyway, so what we have is an example of a calico cat. And what we're talking about is the dorsal coat color, not the ventral. This is autosomal. This is a gene that is autosomal. It has nothing to do with sex chromosomes. Whereas the dorsal coat color is actually going to be a result of what we'll learn, what we'll see the answer is, which is dosage compensation. And so before I go to the next slide, let me just reiterate something. And so if I can reiterate that homozygous males, they only have one X chromosome. So they're only going to have, either orange or black. And so they're going to have uniform coat color. It's going to be one of the two coat colors. It's not going to be both. And so if you remember some of the things we talked about, you might be thinking in the back of your head, is this incomplete dominance? Is this co-dominance? And so technically it's going to be something new, because we are not going to have both alleles being expressed at the same time in a cell. It's going to go by on a cell by cell basis. So it's a little new. It's something different. So Marie-Leon deduced that in heterozygous females, both alleles were active, but not in the same cells. And so, if I had a board, I would be able to show you that what I usually do is I draw like a little embryo and I talk about embryonic development. And the way embryonic development works is, for phylum Chordata, which we are, which cats are, in phylum Chordata, you have bilateral symmetry, meaning, look at yourself, right? You have like two eyes, two heart, let me try that again. Two eyes, two lungs. That's what I meant to say. Two kidneys, and two arms, two legs, right? So there's a rough plane of symmetry going down the center of your body. And so it's like, not like a sea star that has radial symmetry, right? And so we are phylum Chordata, and so we start off with like little pharyngeal slits, and we also have a dorsal hollow nerve cord, and a lot of cells start at midline, so the dorsal midline, and then as they differentiate, they migrate outward from that midline left and right to give us that bilateral symmetry. So that's what's happening here. If we were to look at the back, I'll show you the back of some women's back and backs and then some other things. But if you look at this, what we're having, you can't see the other side. But imagine like on this we have like a cell differentiated, and when it did, dosage compensation occurred early on in embryonic development, where one of the two X chromosomes was inactivated and made into a bar body. So let's say that's the one for the orange coat color containing X-linked allele. So that one's crumpled up and we can't use it, but we have the other one that has the even recessive black allele. And so therefore that cell's going to develop a black coat color, and then it's going to keep that. All successive cells from that one they're always going to have that particular X chromosome as the functional X chromosome and the other one as the bar body. And so you can see it spreads. And so there's a lot of cells from here that all are derived from the original cell that made the choice to use the black X chromosome, the chromosome containing the X-linked recessive allele for black coat color. And then very well, like just right over here, we could see that, okay, well here the X-linked recessive allele for the black coat color was on the chromosome that turned into a bar body. And so you're going to use the orange. And so I guess I should have been doing it right here. So here you see it goes orange. Here you see we went black, right? And that just happens at random. So Leon hypothesized, or the Leon hypothesis of how dosage compensation occurs is as follows. One X chromosome is active in the somatic cells of females, right? We have one that is inactivated as a bar body, one's working, right? Whichever one of these is inactivated is the bar body, the other one's still going. And so that makes it so that you have one functional X chromosome, just like an XY male. So we have dosage compensation. And one of the side effects of this is that the other one that's inactivated is tightly coiled up into a bar body. That means that you're not going to be able to use that allele. And so the inactivated X can be maternal or paternal. It happens at random. And so it doesn't matter. So inactivation takes place early in development and occurs randomly. So it doesn't matter whether you have an X chromosome with a dominant allele or an X chromosome with a recessive allele. You're just going to use one of those per cell at random. And so therefore, you're going to use both of them in the organism, just not in the same cell. So the inactivation is permanent, except in germ cells. Germ cells, the bar bodies are going to be basically you uncrumple the paper, smooth it out, make a copy of it, and then it's a functional X chromosome that can be passed on into gametes. And so all descendants of a particular cell, as I was saying just a moment ago, will have the same X chromosome inactivated. Random inactivation of X chromosome in females is going to be what we call dosage compensation. That is what equalizes the activity of X-linked genes in males and females. Everybody's working off of one functional X chromosome, whether you're XX female or XY male. And so dosage compensation allows certain X-linked recessive disorders to be expressed in heterozygous females. So this is something that you're going to have some test questions about. So let me just say it right now. Let me specify something that's hopefully this will clarify things. In order for dosage compensation to give us a phenotype, right? Like if you're X-linked, if you're homozygous for an X-linked allele, so you have an XX female and she has two of the same copies of a particular gene on her X chromosomes. When you inactivate one, you're going to use, let's say a dominant allele of this gene. When you inactivate the other one, you still have a dominant allele of this gene because she's homozygous for that, right? So if she's homozygous for something on the X chromosome, then we don't see any phenotype coming to us from dosage compensation. That phenotype, as I'll mention in a moment, is called mosaicism. You make a mosaic. And so mosaicism requires an XX female to be heterozygous for an X-linked recessive trait. Or, sorry, just X-linked trait. It doesn't have to be recessive, I'm sorry. She has to be heterozygous for an X-linked trait. And then via dosage compensation, she will end up using one or the other X chromosome in different cells throughout her body. Therefore, she will exhibit mosaicism. So let's put that into bullet points. So dosage compensation allows certain X-linked recessive disorders to be expressed in heterozygous females. So heterozygous females, got to have heterozygous females. Inactivation occurs early on in development, right? So this is very early on before cells have differentiated. Color blindness. We just talked about X-linked color blindness. This is a good example of, I don't know if you've ever heard this phrase. This is an actual phrase. It's an old phrase. I don't know if people still use it or if you've heard it. But have you ever heard the phrase, she has an eye for color? Right? So, she has an eye for color, right? And that might be a phrase that you associate to a particular woman because she does not carry any X-linked recessive color blindness alleles. Therefore, every single cell in her retina is capable of detecting red and green wavelengths of light. Whereas if you had a heterozygous carrier, then some of her cells are wild type. Some of her cells actually are going to exhibit red-green color blindness. So she will have fewer receptors. And when you can really see this at play, so she is not colorblind. She could see red and green, but under low light conditions, she will have a harder time. The heterozygous female will have a harder time basically distinguishing between different shades of green or red. That's where that phrase, she has an eye for color, might have come from, right? At least that's one reading. So I read that once and I thought, okay, I'll buy that. I like the genetic explanation for that. It could be, I'm sure, it could very well be from, derived from some sort of literary work or something like that. But I prefer the genetic explanation. All right, so I mentioned mosaicism. Here it is. So tortoiseshell cats or calico cats, right? They are an example of mosaicism. So female, tortoiseshell cats. If you find a male calico cat, then that is exceptionally rare. You basically have genetic mutations that are leading to that. And so what we have is an example of mosaicism, where it is the byproduct of dosage compensation in a female, XX female, that is carrying heterozygous X-linked alleles. So let's go back to orange versus black. So the dominant O allele is going to basically produce the orange-yellow color, and the recessive O allele, lowercase o, is going to produce the black coat color. So if you have a heterozygous female from just straight Mendelian genetics, you would expect, oh, okay, should be all orange, no black. But in this case, we see orange in certain places, black elsewhere, and that is a result of dosage compensation. And so what we call this type of phenotype is mosaicism. The tortoiseshell cat exhibits mosaicism. And so heterozygous females, so this is all stuff I just said on the previous slide out loud. Here it is in bullet points. Heterozygous females have patches of orange-yellow fur and patches of black fur, and so they exhibit mosaicism. White fur, again, pay no attention to it. It's autosomal. So now keep in mind, mosaicism deals with X chromosomes. So it's not limited to just mice and cats. There are examples. I'll give you some examples of mosaicism in human females as well. All right, so here's an example of mosaicism in human females. It's called Blaschko's lines. So Blaschko's lines are another example of mosaicism. Blaschko's lines are differential pigmentation. This is not anything to do with vitiligo. I usually get that question every single time I lecture about this. So this is not the the long-term loss of pigmentation. This is basically skin pigmentation genes that are X-linked. You can have a dominant allele, you can have a recessive allele, and therefore, because like we talked about, right? So this would be your dorsal, midline. And so as we are developing as human embryos, we then, cell differentiates and then moves outward, right? It moves outward from the midline. And so as it's moving outward, you could see that the different, choice, if you will, the cell makes in which X chromosome it inactivates is going to lead to pigmentation of a lighter tone or a darker tone. And so these are all examples of Blaschko's lines that can occur in human females. Here are some examples. So this little girl, you can see, so it's not just dorsal, right? It could be ventral as well. So you can see that she has, I don't know, I would say that this looks like an example of, I'm no expert. I would go with phyloid pattern. I would think that's probably phyloid pattern. Perhaps narrow bands, but I'm thinking phyloid pattern. So she's an example of phyloid pattern. Here's a better example. So here's an older woman, you're looking at her back, and if you squint, you can see that she lines up almost perfectly with large bands of Blaschko's lines. So B would be large bands. And so, for example, here you can see this kind of flag-like pigmentation area, right? That's kind of like this one. And then you can see the lack of pigmentation, and then you can see another one, right? So you can see lack of pigmentation, another one. You can see lack of pigmentation, lack of pigmentation. There's still some pigmentation up here that goes all the way up her arm. So that would kind of be this one that goes down to this arm. So, I mean, it's just coincidence that she lines up really well. That's why I chose this image with large bands of Blaschko. So now that would be something that you could see. Remember, mosaicism can exist in ways that you can't see as well. And so, like, for example, and usually when I'm lecturing, we talk about the different people in the lecture hall, and I usually talk about the females in the class, and then I switch to the males in the class. Right now we're focusing on the females in the class, and so then I ask at this point, are there any of you out there that have, and you don't have to identify yourself, but that have been battling an area of dry skin your entire life, and it's not eczema or anything like that. It's just for some reason you have dry skin, certain parts of your body, and other parts you don't have any problems, right? That could be because of another form of mosaicism that everybody's heard of because it's so common. It's called anhydrotic ectodermal dysplasia. right? Of course, right? Everybody's heard of that one. And of course, I'm being sarcastic. So anhydrotic ectodermal dysplasia is where you basically have mosaicism of sweat glands. Now, sweat glands are useful for obviously perspiration, for sweating when cooling yourself down, but they also are instrumental in the secretion of the different, the oils and things that you secrete onto your skin in order to lubricate your skin. Lack of those sweat glands means that you don't have that ability, and so therefore you have areas of dry skin. And so that would be an example of a mosaicism that you can't obviously see. And there are other forms of mosaicism, but those are just a couple examples of mosaicism. So mosaicism is not just limited to cats or mice or anything like that. It's limited to, really, it's not limited. It is an artifact of, or it is a possibility, I should say, with XX females that are heterozygous for a particular X-linked gene, so that when dosage compensation occurs, because they need to compensate and only use one X chromosome per cell, when they inactivate one of those X chromosomes to make a bar body, there it is, to make a bar body, then they are going to, in essence, that cell is making a choice. Early on in development, it's making a choice which X chromosome it's going to use. And if, again, that cell is, or this organism is heterozygous for alleles on a particular X-linked gene, then you're choosing to use one over the other. Maybe a dominant versus a recessive. Doesn't matter. You're going to use whichever one is chosen. So what's interesting is that the X chromosome inactivation is still not well understood. So what I mean by that is it's understood that they're randomly chosen. But once the cell differentiates and chooses one, it has to uncrumple that page of paper, photocopy it, and then pass it on to daughter cells in mitosis. Right? These are mitotically dividing cells. These are somatic cells. And then what's interesting is, so it's a fully functional X chromosome in mitosis. It gets replicated, and then you go through the motions, you split, you cytokinesis, and then the exact same one gets crumpled back up again every single time, which is why you saw on our cat large areas of, or here, you have large areas of, so there's many cells that make up these different areas of the body or different patches of fur. It's not one cell, it's many cells. All the progenitor cells, so I should say all the daughter cells of the original cell are going to inactivate the same X chromosome. And so how do the cells, generation to generation through mitotic division, know which of the two X chromosomes to inactivate? Somehow they know, and that we still don't know quite yet. All right, well, let's talk about some other forms of inheritance. So the really only ones remaining are going to be, so we did X-linked dominant, X-linked recessive. Autosomal dominant and autosomal recessive are really honestly the exact same thing, except that you don't have affected phenotype skewed towards one sex or the other. And so autosomal dominant will be 50-50 distribution between males and females. Everybody's affected. At least one person's affected every generation, right? Autosomal recessive is 50-50 distribution, and then you might skip a generation here or there. Now, that brings us to the only two left are Y-linked and mitochondrial inheritance, or maternal inheritance. And so since we're on Y-linked, let me just quickly say maternal inheritance is mitochondrial genes. In our mitochondria, we have certain DNA. And so mitochondrial inheritance is nice and easy. Y-linked and mitochondrial are the two easiest. Mitochondrial or maternal inheritance is mother to all children, mother to all children, mother to all children. So you just follow up the maternal line. That's it. Fathers never pass on mitochondrial genes because they only provide the sperm cell, not the egg, which contains mitochondria, which is mitochondrial DNA. So that's why it's called maternal inheritance or mitochondrial inheritance. Mother to all offspring, that's it. And then Y-linked is somewhat of an example of Murphy's Law. So if you're not familiar with Murphy's Law, basically, sarcastically, we could say, because Y-linked inheritance is the easiest to identify in a pedigree, you're probably never going to see it, right? That would be what I mean by Murphy's Law. And the reason why is because, okay, so Y-linked is the easiest to identify in a pedigree because it's basically male, so father to son, that's it. So just like maternal inheritance is mother to all children, Y-linked inheritance is father to sons. And that's it. And that's because only males carry Y chromosomes, and so therefore the traits passed are directly from Y, on the Y-linked traits are passed directly from father to son. So father to son, father to son, father to son, father to son, right? Father to son, father not to daughter because he's giving his X chromosome to his daughter and his Y chromosome to son, and then he's giving his Y chromosome to his son, not to his daughter. Same thing there. Same thing there. And so the problem with this is it's super easy to recognize because don't get it confused with X-linked recessive. That X-linked recessive pedigree I showed you a little ways back had only males affected, but remember, it skipped generations. And it passed through a female, right? So like it would go from, like from father to daughter, and then the son was affected again, right? And so that was how you knew that it wasn't Y-linked because it's not going to go from father to daughter to son. It has to go from father to son, father to son, father to son. And so the other, the only thing about this is it's super easy. Y-linked is super easy to recognize in a pedigree. And of course, that means that it's also going to be the most rare. You're almost never going to see it. And that's because, remember, the Y chromosome is so small. It has so few genes that you're really not going to run into this very often. So this unique pattern of inheritance is nice, but there are less than 50 possible Y-linked traits discovered to date. So when I say possible, that means that they haven't been characterized. So it could be far less than that. Really, 20 to 30 Y-linked genes are actual genes that we know about. The remaining genes are gene candidates that nobody's really checked yet. Remember, you kind of identify stuff. Once they sequence the human genome, you just run an algorithm search and you look for what appears to be a gene. Then it gets a designation, and then it's taken a while for all of us to, we haven't even come close to looking at all the different genes, right? And so, you don't really see Y-linked inheritance very often. Okay, so now it's very important that, I hate to do this to you, I'm sorry, but with genetics, basically the more you learn, the more levels of complexity we just add on to things. And so what you have seen thus far has been dominant traits show in the phenotype, recessive traits get masked by dominant phenotype or dominant traits and don't show in the phenotype in a heterozygous individual, and homozygous recessive show their phenotype in the organism, right? Well, that is kind of Mendelian dominance. That is basically saying, oh, the dominant allele gives the dominant phenotype. The recessive allele doesn't if there's a dominant phenotype there, but if there isn't, then it shows the recessive phenotype. But here we go. Sometimes genotypes don't always produce the expected phenotype. And what that's called is incomplete penetrance. Incomplete penetrance is this extra level of complexity where, and I've said this way back in the beginning of the course, right? It's not all genetic. And so you could be homozygous dominant for, I don't know, a lethal allele, but so you have two copies of this lethal dominant allele, but you're alive and well and kicking, right? And so that's because their phenotype is not penetrating through and showing, and therefore in that case being fatal. So it would be incomplete penetrance. So penetrance is a number. Penetrance is the percentage of individuals having a particular genotype expressing the expected phenotype. So in Mendel's pea plants, he's never ran into incomplete penetrance. He had 100% penetrance. So if it was dominant, homozygous dominant, or heterozygous with that dominant allele, that was the phenotype 100% of the time. But if you keep going and you look at all sorts of other genes, especially in human beings, you will find that there are varying levels of penetrance. So for example, if 8 out of 10 people have a particular dominant allele, so let's say 8 out of 10 people, let's just make them heterozygous. So they have one dominant, one recessive allele. And so 10 people. And then 8 out of the 10 show the phenotype and 2 don't. Well, that means that it's 80% penetrant. So here's another one. If 38 out of 42 people with the allele for polydactyly, meaning an extra digit, right? So polydactyly. Sometimes I use polydactyly. Sometimes I use camptodactyly. Camptodactyly is very similar. It's when you have an immovable bent little finger, right? So your pinky finger is going to be, sometimes it could be like this. So you would have the correct number of fingers. You wouldn't have an extra finger, but your pinky finger would be like kind of just curled up and really small, and you couldn't really use it. Sometimes it's like a little nub. That would be camptodactyly. Polydactyly, as the name implies, right, is an extra digit. So you can see 1, 2, 3, 4, 5 fingers, 1 thumb. So basically an extra digit on this hand. So if 38 out of 42 people with an allele for polydactyly are polydactylous, well, then that works out to a 90% penetrance rate. And I can tell you, I remember this image, using this image a long time ago when I first saw it. I put it into my slides, and this was before my little girls were born. And then when they were born, they were so small and just all wrapped up. And I mean, they were full. It's not like they had to go to the NICU or anything, but you know, they're little babies, and they're all swaddled and wrapped up, and we're in the hospital, and I'm sleep deprived, and their little hands were like grasping onto their little blanket that they were swaddled in. So I did it wrong, basically. And so. And they kind of, I just kept thinking of this picture, and for some reason I just kept counting their fingers, because there looked like they had like 20 of them, because they were so little. It was hard to determine, in between the little fingers, like which one's which. And I remember saying to myself, why am I counting? I counted already. It's fine. They have regulars, so why am I counting again? But I was just, I was very sleep deprived. And so every time I saw their hands like that, I was just like, that doesn't look right. And so I kept counting it over and over again. And they're still the same. The number didn't change, surprisingly enough. All right. So now, expressivity is a different thing. So there's penetrance and expressivity. They kind of go hand in hand. Penetrance is the yes or no. It's the percentage. So 90% yes, 10% no, right? Expressivity is, okay, if you're in the yes category, to what extent, right? The degree to which a trait is expressed. It's related to penetrance. You have to be a yes in order to have any sort of expressivity. But if you do, so like, for example, here's camptodactyly. Think of it as, okay, let's look at expressivity in terms of which hand. So there could be like your left hand, there could be your right hand. You could have camptodactyly in your left hand, your right hand, or you could have a higher degree of expressivity and you could have it in both hands. You could have an even higher degree of expressivity and you could have it in your hands and your feet, right? So basically expressivity is like, okay, so you answered the yes or no, and now, okay, so to what degree do we have a trait being expressed? And so some individuals show affected phenotype for camptodactyly, polydactyly in their left hand, some in their right hand, some in both hands. This is a pedigree that I kind of butchered. This is not a proper pedigree, right? So what I did was basically just use it to say, okay, well, this is in the right hand, this is in the left hand, this is in the right hand, this is in the left hand, this is in the right hand, this is in the right hand, both hands, and then left hand, right? So that means that this is a higher degree of expressivity than all the rest of these. So that's just an example of, what we're talking about with expressivity. So I won't give you any pedigrees that look like this, because that's just all sorts of wrong. All right, so others, there are other types of variation in gene expression. Others have a wide range of phenotypes or show delayed expression. Like, say, for example, that lethal dominant allele that I mentioned before. What if it's lethal dominant later in life, right? It has a late onset. So your gene expression changes throughout your entire life. So then when you hit, like, let's say, a relatively early age for this, so let's go with like 40. When you hit your 40s, then you actually start to see the effects of that lethality, people dying off at a much earlier age than half their life expectancy. So what happens is you have variations in phenotypic expression that's caused by a lot of different factors. And so I just mentioned age, right? So our gene expression changes constantly. Don't think of it as just like, okay, when you're babies, stuff's happening. Then when you hit puberty, a lot of stuff changes. And then as you get older, maybe, I don't know, you think of like menopause or something like that. No, your gene expression is changing throughout your entire life. And so there are different aspects of that are good and bad. If you carry the Huntington's disease alleles, right? So these are lethal dominant alleles. So one allele enough, lethal dominant allele. And Huntington's is a devastating disease. It's a neurodegenerative disease, and basically what happens is, just imagine like your mother. You're getting. You're fairly, old. Let's say you're at least in your 20s, and so your mother. You have an argument with your mother. You've argued with your mother before, but this time And this is not meant to be a joke or anything like that, but this time something is very wrong. Something's very different. Like, for example, she gets so angry, she, comes at you with a knife, right? Like, that's not a normal or argument you've ever had with your mother before. And so then you clearly know something's off. That's usually a trigger when usually family members realize something is very wrong. Violent mood swings, and then all sorts of just changes to the individual's psyche. That happens with the onset of Huntington's disease. And so at that point, there's a gene that you can look at, and you can see how many different CAG repeats, so CAG bases, we'll get to those later, repeats are in that gene. And if you have a certain number of repeats, then it's a lethal dominant allele with 100% penetrance. So you in your 20s, like your mother gets diagnosed and she has the beginnings of the onset of Huntington's, that was just devastating to you because it's lethal. Like, her time is very limited at that point. And then the thing is that it's lethal dominant. So the family gets tested, and then you find out, for example, that say, for example, you're a carrier of this lethal dominant allele. You know that basically your life expectancy has now been changed. It is now far less than what you originally thought, because later in life you would expect to also have this, because unfortunately, that's why I say it's a horrible, debilitating disease, and the diagnosis is usually really, really bad, because it's not just for the one individual, it's usually for at least a few other family members as well. So, of course, you could also have genetic interactions where your genes interact with one another, or they could interact with the environment, right? So interactions with the environment could also affect variations in gene expression. So don't forget that it's not all genetic, right? So, like, for example, a separate gene that shares the exact same name just by coincidence with Huntington's disease is the 5' HTT gene. The 5' HTT gene, HTT. So we're not talking about Huntington's. In this case, we're talking about clinical depression. And so there are individuals that carry this mutated gene, the 5' HTT gene, and they are predisposed for clinical depression or depression if there is a trigger. Right? So interactions with the environment. So usually there's some sort of traumatic experience that kind of triggers the expression of this gene. And that contributes to their depression. But if you're fortunate enough not to have a truly traumatic experience, or at least go through most of your life without that traumatic experience, then you may not actually trigger the expression of this gene. And so that would be an example of an interaction with the environment. So keep in mind, the mere presence of a gene does not guarantee its expression. It is not all genetic. And that is a common misnomer. That's why this bullet point is in all red. I cannot stress this enough. You are in a genetics class. You are now far more educated in genetics than the average individual. So remember that just because you have a genotype, just because you have a gene, doesn't necessarily mean that you are going to have the dominant, if it's a dominant gene, it doesn't mean you're going to have the phenotype. Or even if it's recessive, it doesn't mean you're going to have the phenotype. And this is both good and bad, right? So this could be, like if you have a beneficial gene, a functional gene, and the lack of that gene causes a disease, and you have the functional wild type gene, but yet you still have the disease, that's unfortunate. That's, incomplete penetrance of that gene that you need, and it's not functioning, and so therefore you have the disease. Alternatively, if you are diagnosed with a particular, like for example, Huntington's is a bad example, 100% penetrance. But let's say you're diagnosed with you find out that you are a homozygous dominant carrier for some other lethal dominant alleles, 2 of them. You have 2 of them, but it's only 30% penetrant. Well, then now you're in good shape. Now hopefully you're not in the 30% class, you're in the 70% class. And depending on if it's late onset or not, if it's early onset and you're still alive, well, hey, you're definitely in the 70% because you just don't have the penetrance of those particular alleles to cause that lethality, which is wonderful. So now another variation in gene expression is caused by gene interaction. I didn't quite mention what gene interaction was, genes interacting with other genes on that previous slide. So let me flesh it out here. So frequently, alleles of a gene exhibit independent assortment. We've talked about that before, but they do not act independently in their phenotypic expression. So this is called pleiotropy. This is when you have multiple alleles contributing to a phenotype. And so here's what we have. We have the figure from your textbook, I believe, that contains the most elusive bell peppers I've ever seen in my life. Now, I've seen red bell peppers, and I grow stuff. I have a garden and all that. I'm I'm pretty good at it too. And so what we've got is red bell pepper, sure. Cream bell peppers. Now, I've polled many quarters worth of classes, and I've been teaching this class for a while, and each class has around 90 students or so in it. So we're talking like thousands of students at this point. And of the thousands of students, when I ask, I usually get maybe, total of all of them that I've ever polled, I've had about like 20 students total verify that cream bell peppers do exist. So I still don't think they exist. But no, I'm just kidding. So, okay, so we have cream bell pepper. So we have our red bell pepper. Red bell pepper is going to be homozygous dominant for Y, homozygous dominant for C. Cream is going to be homozygous recessive, homozygous recessive. We're going to do a cross, and we're going to say, okay, well, that makes sense, right? So this is following Mendelian genetics so far. So, you're going to get all heterozygous for the F1 generation. You get red bell peppers. Okay, I buy that. Then you do a self-fertilization, right? Or you just cross your heterozygous with another heterozygous. Very, very similar to what we saw when we did a dihybrid cross when we were talking about transmission genetics. And in this case, you get 4 different phenotypes. You get red bell peppers, which are readily available. You get orange bell peppers, even though that to me looks yellow. I would call that a yellow bell pepper, but I'll give them that one because, orange bell peppers do exist. I've seen those, right? You can go to Costco and you get a pack that contains 1 red, 1 yellow, and 1 green. And sometimes it contains 1 red, 1 orange, and 1 green. Probably information you don't need. That won't be on the test. Then you get, okay, that is clip art of a pumpkin. I don't care what anybody says. That is clip art of a pumpkin. That is a pumpkin. And first off, it's a pumpkin. Second off, calling it a peach bell pepper is a stretch because I would say that that's a freaking orange bell pepper and that's the yellow bell pepper. I don't know who came up with this, but if I was the graphic artist in charge of this, I would be, fired. But anyway, what we have is, okay, so let's just go with it. Let's pretend that this is a red bell pepper. I'll buy that. This is, a peach bell pepper pumpkin. This is an orange bell pepper yellow. And then this is, a unicorn. So basically my point, though, is that you do get different outcomes depending on which alleles are mixed with which alleles. So, regardless of my tirade about bell peppers, what we see is gene interaction. We see that it depends on which allele of which gene and what combination you have, that they interact with one another to give you phenotypes. And there are different phenotypes here. There are new phenotypes, right? We saw the red and the cream, but then we also see peach and orange. Can't say, that is just, it's a pumpkin. All right, so alternatively, there is also I gotta collect my thoughts here because I'm still fuming about that. That's a pumpkin. All right. So alternatively, one could one gene could also mask. So this is a gene interaction where they're kind of working together to give you a phenotype. You could kind of have the opposite of that, where you could have one gene can mask the effect of another gene at a different locus throughout anywhere in the genome. That's called epistasis. Epistasis would be, that's one example of epistasis. And so epistasis would be like, for example, imagine an autosomal gene shutting down a gene on the X chromosome. And I just took those two, it could be an autosomal gene shutting down another autosomal gene on a different chromosome. Doesn't matter. So when one gene interaction basically halts the expression of another gene, that's a gene interaction, but that's a specific type of gene interaction where you're kind of like negative. So that would be an example of what's called epistasis. All right, so epistasis is important to remember that it involves different genes, not alleles of the same gene as in gene interaction. So here we have two genes, so there are different genes, but it depends on what alleles we have of each gene. So the alleles play a role as well. Whereas epistasis is just one gene shuts down another gene regardless of what alleles are present or anything like that. There are other things to consider as well in basically gauging phenotypes. And so what we're going to talk about now are two of these, and they are sex-related or, so I should say sex-specific or sex-influenced traits. Both of these are sex-related phenotypic effects, and so that's why that's the title of the slide, right? Sex-related phenotypic effects. But one of them is sex-limited, limited to one sex or the other, and then one of them is sex-influenced. It's like, say, dominant in one sex and recessive in the other sex. So in some cases, phenotypic expression of a trait is going to be different in the male as opposed to the female, or in the female as opposed to the male. And so let's start with sex-influenced traits. Sex-influenced traits are expressed in both sexes. So sex-limited, as you'll see in a moment, is limited to one sex or the other. Sex-influenced, very important you remember, it's expressed in both sexes, but differently in males and females. And so it is controlled by autosomal genes that are usually dominant in one sex, recessive in the other sex. And so this illustrates the effect of hormonal differences, for example. There are many different ways in which we can look at this, but let's look at hormonal differences. So this illustrates the effect of hormonal differences on gene expression. Different hormones stimulate different genes, or different concentrations of hormones stimulate different genes. For example, if you've ever heard it referred to as male pattern baldness, that's actually not accurate. It is pattern baldness. And pattern baldness is seen more often in males, which is why people call it male pattern baldness, because it's autosomal dominant in males, but then in a female, it's recessive. And so if you've ever seen like different, I don't know, I haven't seen any of these in a long time, but commercials like for Hair Club or Bosley or Rogaine or different types of regrow your hair. I think Keeps is one of them. Basically, there are different things that different ways in which you can try and stimulate hair growth. So they're marketing to mostly males, because it's autosomal dominant in males, and so mostly males are the ones that are losing their hair. However, if you look closely every now and then, so certain ones, I can't remember, I think it was like Bosley or something like that. I remember seeing that they're like, showing the before and after. There's like the unhappy face with the bald head, and then the way better lit, smiling, happy guy with more hair. And they're paging through those as they're talking. And then they always show like one lady, right? There's always one lady, one little old lady that's like, got very little hair on the left. And then she's super happy and has like, a lot of gray hair on the right. And so that's because it is actually, so it depends on what type of medication and stuff. If you've ever seen I lecture about Propecia in a different class, one of my biochemistry classes. And wow, if you've ever seen the warnings for Propecia, it's a drug that people use to grow hair. I still have my hair, which is nice, and when I go bald, I'm just going to shave it all off. I don't care. But basically, Propecia is this drug that you can use to try and regrow hair, and like the handling of it is amazing. There's all these warnings for women to stay away, right? So like pregnant women should not handle Propecia. Pregnant women should not even use blah blah. They say like you should wipe down the sink and everything in the bathroom if the male who like took it, before the female comes in, especially if she's pregnant, etc. But the point is, that's an example. So pattern baldness is an example of a sex-influenced trait. You see it in both, but it's autosomal dominant in males, and it's recessive in females. And I guess one last thing to say about it is that it's related to a genetic variant in an X-linked male hormone receptor gene. So if since we were talking about the females in the class, and like things like anhydronic ectodermal dysplasia, or Blaschko's lines, let's talk about the males in the class. So if you want to know what's the likelihood of you losing your hair, so it's going to be passed via your mom because it's X-linked. So you're going to get it from your mom, but obviously, it's recessive in her, so she's probably not going to, be bald. And so you want to look to remember, she got one of her X chromosomes from her dad. So look to your maternal grandfather to get an idea, not a guarantee, but to get an idea of your likelihood of going bald. Right? So like if you care and you don't want to go bald, and your maternal grandfather had full head of hair, or has a full head of hair, then you're in good shape. If he's bald, doesn't mean that you're going to go bald, but it certainly increases the chances of you going bald. Whereas if you look to your paternal grandfather, it doesn't matter, because you got your Y chromosome. So XY males, you got your Y chromosome for your dad. So that, what he has in terms of his hair, your dad or your paternal grandparents, that doesn't matter, because we're talking about something that's X-linked. So I guess you could say you get it from your mom, or at least your mom's side of the family. All right, now let's talk about sex-limited traits. Sex-influenced, both sexes, usually like dominant in males, recessive in females, or dominant in females, recessive in males, like that. Sex-limited traits are pretty straightforward, right? So like prostate cancer, that's pretty sex-limited because men have prostates and women don't. Ovarian cancer, it's pretty sex limited. And so it doesn't have to be that straightforward, though. So sex limited traits are inherited by both sexes, but normally expressed only in one sex. And so they produce a phenotype only in one sex. Now, the examples I gave were they were physiologically sex limited. Right, so prostate versus ovaries, that sort of thing. So let's actually talk about something that could possibly occur in both male and female, but is effectually sex limited. So Duchenne's muscular dystrophy is basically, and it's an X-linked disorder. It's basically sex limited. And so let me explain what I mean by that. So there are always the examples that are purely sex limited that are easy to come up with because, if you talk about the prostate, you're talking about males. If you talk about the ovaries, you're talking about females. Then there's some other examples as well. And so those are going to be examples that are absolutely sex limited, right? And so if there's some sort of inherited trait associated with the prostate or ovaries or something like that, or testes, then you're talking about sex-limited traits. But here's one that's a little bit more subtle. So Duchenne's muscular dystrophy is, like I said, X-linked disorder, basically sex-limited. And if you're not familiar with Duchenne's muscular dystrophy, what that is it's basically it's a genetic disorder in which your dystrophin protein is not functional. And your dystrophin protein is very important in anchoring your muscle fibers to cytoskeletal components so that when you contract your muscles, they have something to hold on to. And so that creates the motion and it doesn't rip apart cells. When your dystrophin protein is not properly connected to your cytoskeleton, when you contract your muscles, you literally tear apart your muscle cells. And so it's an extremely debilitating disorder. And so it affects 1 in 3,500 males. And these males are going to have a very hard time with motor control, speaking, even, anything you use your muscles for. So bowel movements, whatever it is, those are all going to be quite the challenge. And you're destroying your muscle tissue every time you contract your muscles. But this tends to kill off, unfortunately, it tends to be fatal sometime after the teenage years because you're also rapidly growing up until that point. Once you really get to an adult age, then you stop growing as quickly, so you can't regenerate your muscle as quickly. So when you're destroying your muscle, you're also growing new muscle because you're rapidly growing. And then once you get to a point where your growth levels off, then it's just deterioration at that point. And so these individuals, they don't usually live, past their 20s. And many die long before then. So it affects 1 in 3,500 males, and it's essentially fatal. It is possible. It's excellent. So it is possible to get a homozygous recessive female, and so it affects 1 in 50 million females. So look at the number difference there, right? So that alone tells us that you're basically sex limited. It's basically you see it in males. You almost never see it in females. And the reason why you almost never see it in females is because, what we just talked about is that the male is going to usually, unfortunately pass away before reaching a sexually reproductive age. And even if he did, so I mentioned 20, so okay, so let's say that he did reach a sexually reproductive age, he's going to have a very difficult time reproducing, right? The muscle contraction necessary for ejaculation, everything is going to be extremely, difficult for him to properly father a child. And so he is not going to contribute one of those alleles. And so 1 in 50 million means that we're talking about like maybe a heterozygous carrier that picks up an extra mutation and then ends up being, having Duchenne's muscular dystrophy. It's just unlikely, right? So basically all the scenarios are stacked against it being prevalent in females as opposed to in males. You just have to have one copy of this, the DMD gene, and the mutant DMD gene, and then you have Duchenne's muscular dystrophy. So, like I said, affected males die before reaching reproductive age, or even if they have reached a reproductive age, they're usually going to it's going to be very difficult for them to reproduce, if at all. So, affected females are extremely rare. Now, one other thing that I want to mention before we talk about a little bit more of the variation is imprinting. I don't have a lot to say about imprinting because we don't know a lot about imprinting. Imprinting is pretty interesting. Imprinting is a phenomenon in which gene expression depends on whether the chromosomes are paternally or maternally inherited. Now, somehow the cell knows whether its chromosomes came from males or females. This is why, just recently in the news, I think it was 2018, I think it was, there was this news article about a three-parent baby, but it wasn't really a three-parent baby. And so same-sex couples, for example, no matter what we can do in a laboratory setting, they still can't have a child through a lot of manipulation in the laboratory. They still can't have a child with genetic contributions from just the two of them because of imprinting, right? You can't take so males have X and Y, right? So you can't take a set of autosomes from a male donor and then let's say his X chromosome, and then a set of let's say they want to have a daughter, right? And forget this is okay. So I'm not talking about the ethics of this, right? So this has been done with other organisms, not humans. So in a laboratory setting, let's do it that way. So we have an organism, an experimental organism, and then chromosomes are taken from a sperm cell so that you have a set of autosomes and then one sex chromosome, set of autosomes from another sperm cell from a different organism of the same species, and then one sex chromosome. You put them all together into a enucleated egg cell. So you basically, you have to use some sort of egg cell, and so you take the genetic complement of the egg donor out, so it's enucleated. You put those two nuclei, the sperm cell nuclei, in there, they fuse, and now you have basically a diploid zygote whose chromosomes are all derived from, they're all functional, they're all healthy chromosomes, but they're all derived from male parents. So it's a male offspring, right? And then somehow the cell knows, and it's an inviable diploid zygote. It's an inviable embryo, and you'll have a failed pregnancy. And so you could try with all sorts of different combinations, but somehow the cell knows, and that is what imprinting is. And that's all I can say about it. It's fascinating to me because it's one of those questions that are still largely unanswered. Very, poorly understood at this point. Okay. I think now what we need to do is to take into account, I've basically told you all these different instances and how it's not all genetic, right? Well, now let's talk about, so Mendel had very, very specific outcomes with his pea plants, either tall or short. Remember, I kept talking about how binary it was, either yellow or green. I joked about, either smooth or wrinkled, never sminkled, right? And so it's one of those things where he had this or that. Those are what are called discontinuous. It'll be in the bullet points. The discontinuous traits or discontinuous phenotypes. Whereas humans and other organisms have a tremendous number of continuous phenotypes or continuous distribution of phenotypes. So that's where we're going with this. So in pea plants, for example, one gene pair controls height, and you have either tall plants or short plants. You can count up the number of tall plants, you can count up the number of short plants, and you can make a bar graph where you have your tall and your short. And so in humans, on the other hand, if we were to look at height, if we were to look at heart disease, if we were to look at a number of different things, we have different types of traits. So let's start with heart disease. Heart disease is a very complex trait, and that's an actual term. So there's going to be complex traits, multifactorial traits, and polygenic traits. So a complex trait is determined by several gene pairs and non-genetic factors, environmental factors for which we cannot quantify. So that's complex. Think of all the different things that can contribute to heart disease, not just your genes, but your diet, your access to food, your exercise level, all sorts of different things, right? So heart disease would be an example of a complex trait. And so complex trait is basically a trait that is polygenic. There are multiple genes, several gene pairs that contribute to its phenotype, and an unquantifiable amount of environmental interaction. So tall and short pea plants, as I said a moment ago with Mendel's pea plants, those are discontinuous characteristics. There are two distinct phenotypes, no overlapping classes. In humans, however, let's look at tall or short. So human height is not, you don't count up how many tall people and how many short people there are. You have to measure it. That's because a height in humans is an example of a continuous characteristic or continuous characteristics. And so continuous characteristics are going to be described as a distribution of phenotypic characters from one extreme to another in an overlapping fashion. Right? Think about that. We're going to have like, the shortest person ever measured, and then you're going to find people of various height all the way, like overlapping heights, right? So there's, let's cut it off at an inch. So, there's a difference between someone that's 5'6" and 5'7". But of course, there's going to be people that are, like 5'6" and 3/4, 5'6" and a half, 5'6", really just exactly 5'6". So there's really, if you look even closer, there's overlapping phenotypes. So if you have a distribution of phenotypic characters from one extreme to another in an overlapping fashion, you have what's called a continuous distribution of your phenotypes or continuous characteristic. And we can see that here. So this is, I took this from a different textbook, but this was a genetics class, large lecture. And so they put the males in blue shirts or blue outfits and the females in red. And they basically used some bleachers and they distributed people based off of their height. And so you can see that we start at 5'3", and so you can see he's not very happy about that. And then it goes all the way to 6'4", and he actually turned out to be, it appears, I think it says 6'5", or 6'5", or 6'6", one of those things, probably 6'5". And so what we have is, you have your outliers, and then this is supposed to represent a bell curve distribution, right? So 5'10" obviously is the most common height for males in this particular genetics class. And then here, actually, the women, since there's so many more of them in this particular genetics class, they make up a nice bell curve distribution. It looks a lot better, right? Because it just, there's just more of them, and so that smooths out our curve. And so that is an example of, you could see, a distribution of phenotypic characters from one extreme to another in an overlapping fashion. It's not like we have any gaps, right? We don't have any gaps in between here. So there are lots and lots of people of different height, and so you have to measure their height. That would be a continuous variation or continuous characteristic. So the traits show continuous variation, as I said, and traits showing continuous variation are often controlled by two or more gene pairs. You got to have two or more gene pairs because you need to have a lot of different. If we just had one gene pair and there were only two alleles, well, then you'd have this or that. You'd only have two characteristics. So there would only be two phenotypic classes. But here we have a lot of phenotypic classes, and so therefore there must be multiple genes and multiple alleles of those genes that contribute to give us this overlapping fashion that gives us this bell curve distribution. And so polygenic traits are traits that are controlled by two or more gene pairs. So let's make a list. What I usually do is on the board, I make a list of these terms. And I start with polygenic. Polygenic traits are controlled. These are traits. The phenotype is controlled by two or more gene pairs. That's it. No environmental influence whatsoever. This is one of those times where if it's polygenic, it is all genetic. So it's going to depend on what you have, what alleles of what genes you have, because it is controlled exclusively by the genes that you have. So the example I'll use is eye color. Your eye color is determined by a number of different genes, and that's it. Right? It doesn't matter if when your mom was pregnant with you, if she ate a whole lot of spinach because she was trying to give you green eyes, right? That's not going to work. And you can see how silly that sounds. I feel silly just saying it. And so that's not how this works. It all depends on what alleles of what genes you have for eye color. So let's do a simplified, example of eye color. This is not exactly how eye color is determined because it's actually much more complex. It requires up to 8 different genes. We're just going to do 2 genes. So eye color is a polygenic trait. And so let me say this one more time. A polygenic trait is the, what we use to refer to a particular trait, so a phenotype generated by 2 or more genes. That's it. 2 or more genes generate a particular phenotype. no environmental influence or anything like that. It's just purely genetic. That's an example of a polygenic trait. So it could be 2, it could be 20, but it's a polygenic trait. And the thing about polygenic traits, or any sort of polygenic trait, so we could say a strictly polygenic trait, or we could say a multifactorial or complex trait. Multifactorial is when you have a polygenic trait with some defined amount of environmental contribution. And a complex trait, like heart disease we saw earlier, is when you have a polygenic trait with an unknown environmental contribution. So any one of those, basically, as the number of loci controlling a trait increases, the number of phenotypic classes increase. So eye color is controlled by the OCA2, the HERC2, and multiple other genes. I believe there are 8 genes total that are involved in the determination of eye color. But for the sake of simplicity and for this example, we are going to keep it very simple. We are going to just say that there are 2 genes involved in eye color, and we are going to, because it is a hypothetical, we are going to say that the eye color only goes from dark brown to a lighter brown to a light blue to a dark blue. So we're not paying any attention to hazel or green or any sort of other eye color or any of the other, like multiple other shades of brown or blue. And so that's just so that I can keep this within a manageable number that I could actually work out on a board in a normal lecture. And so what's going to happen is, let's start with 2 loci, right? So now I have to do this aloud. So if you want to write this down as I'm going, that'll probably help. So I'll give you, hold on a second. Let me just say this. As the number of classes increases, there's going to be less phenotypic difference between each individual class, or the individual classes. Okay, so you can see that here what we have is, you can see 1, 2, 3, 4, 5 different phenotypic classes. So that would be this, 1, 2, 3, 4, 5 different phenotypic classes. So that means that we have 2 loci. How do I know that? Well, let's keep it simple. Let's call it the A and B genes. So there are 2 genes. We're calling this a polygenic trait. And for the sake of simplicity, we're just going to have 2 genes. Now, we are diploid organisms. So if we have the A gene and the B gene, that means that we have 2 loci for A and 2 loci for B. So you basically have 4 blanks to fill in. And then, again, let's keep it really simple. Let's just say that there are only 2 alleles for each one of these genes. There's the capital A, lowercase a, or the capital B, lowercase b. So let's say that the dominant alleles produce the pigment and the recessive alleles produce less pigment. And so you go from all pigment containing to less pigment. So then if we look at our 2 spots, right, our 4 blanks to fill in, we could have capital A, capital A, capital B, capital B. And that's it. The all dominant, there's only 1. And so that would be, this percentage of individuals. Then we're going to see there's a higher percentage of individuals could have this phenotypic class, because this would be if you had 1 recessive allele. So if you had 1 recessive allele, think about it. You could have capital A, lowercase a, capital B, or you could have capital A, capital B, lowercase b. So that's why you have more possibilities that lead to this phenotypic class. Then we have the one that has the most possibilities. So this would be if you had 2 recessive alleles. So you could be capital A, lowercase b, lowercase b. You could be lowercase a, capital B, or you could be capital A, lowercase a, capital B, lowercase b. So that would be 3 different possibilities. So that's why you see the most individuals in this phenotypic class. And then, of course, now we have 4. You could look at it as 4 recessive alleles. I take that back, sorry. 3 recessive alleles, right? This 2 recessive alleles. So this is 3 recessive alleles, or you could look at it as having, 1 dominant allele. So you have 2 classes, right? You have capital A, lowercase b, or lowercase a, capital B, lowercase b. And then you have all recessive, lowercase a, lowercase b. And so that would be how this works for 2 loci. And then if you had 3 loci, well, then you would have 6 blanks in a diploid individual to fill. And so you can see that we're kind of spreading out our distribution of phenotypic classes. And there's going to be less difference between them, because now the differences are going to become more and more minute. 4 loci, right? And then, of course, as I said, with human eye color, you actually have 8 genes believed to be involved in human eye color determination. So that's 8 loci. And so that's going to be a lot. I mean, that means 16 blanks to fill. And so you have a lot of variation, which is why we have so many different types of, just brown alone, so many different types of eye color. Or different shades of brown. And then, of course, you have all the other colors as well. So all the blues, all the greens, etc. And so you end up with quite a bit of variation. But in all cases, none of these eye colors, and it's kind of creepy how they're always looking at you, no matter where you are, they're looking at you. But none of those eye colors, or any eye color for that matter, none of the eye colors are going to be determined by any sort of environmental factor, right? It's all purely genetic. So that's a purely polygenic trait. Multifactorial traits, on the other hand, are polygenic. They're controlled by 2 or more gene pairs, and that's a big and show significant interaction with environmental cues. So like, for example, height. Human height would be an example of a multifactorial trait. We have a pretty good idea of what genes are responsible for height, and we have a pretty good idea of what you need to eat in order to, grow to your genetic potential for height. Not everybody makes it to their genetic potential for height. And so you can imagine, like, for example, say we have a malnourished individual that doesn't have proper access to nutrition. They're not going to reach their maximum genetic potential for height. Regardless of what genes they have, they're not going to get as tall as they could be, because you do need certain things in order to grow, right? You've heard that milk does a body good. So each gene involved in multifactorial traits are basically inherited in Mendelian fashion. But then, of course, there's going to be that environmental interaction. And so the interactions of the genes and the environment are what yield variations in the phenotype, so variable phenotypes that don't show Mendelian ratios. And so you get a distribution of phenotypes that, like we saw, gives us a tremendous number of different phenotypes. And so that ends up being something like in the human height example I showed you, where you get a nice bell curve distribution. And so in humans, most traits are multifactorial, not polygenic. So polygenic alone is just 2 or more genes, no environmental influence. So multifactorial traits are polygenic and you get the environment. So most traits are multifactorial in human beings. And then we have the complex traits. There's not much I could say about complex traits. They are complex. We don't know enough about them. Think of hypertension, which is high blood pressure. Think of all the things that can contribute to your high blood pressure. So you could have familial hypercholesterolemia, which is a genetic disorder in which you have an imbalance in your cholesterol metabolism. So it causes hardening of the arteries because cholesterol is spilled throughout your vasculature, and that atherosclerosis makes your arteries hard so that they don't flex as much, and so you end up getting back pressure, and so your blood pressure goes up, etc. That's one way in which you could have high blood pressure. Alternatively, you could be, say, for example, stuck at home during a shelter-at-home order with your family, and a certain family member just does everything in the world to annoy you, even though they're not really doing anything to purposely try to annoy you, and that could affect your blood pressure, right? That's pretty complex. How do you know which genes are involved in that, right? So you know that certain genes are involved, but you also know that certain environmental cues are also involved. Obesity is another one, right? We know of the obesity gene, right? The leptin hormone gene, and so there's that, but then of course there's also eating habits. There's also access, right? So like if you live in an urban area in a food desert where you don't have access to proper nutrition, that could contribute to obesity. If you're in an area of high crime and you don't feel safe exercising or going out or anything like that, then that could lead to sedentary lifestyle. That could lead to obesity. And then of course the genes that you have are also going to play into that. So there's all sorts of examples of, and we already talked about cardiovascular disease. There's all sorts of examples of complex disease. Complex diseases, or complex traits, I should say, are polygenic and have an unknown amount of environmental influence. We know that the environment is involved, but we don't really have the means to quantify that yet. And I stress the yet because, a while ago, height was in the complex category. And now most people kind of put it into the multifactorial category because we have a pretty good idea about what nutrition is required in order to, and the nutrition being the environmental influence, is required for reaching your maximum genetic potential for height.